Hello friends, welcome to entranceadvisor.com. Today I am going to talk about another drug that is going to be used in the treatment of the COVID-19 or the novel coronavirus, which is under trial. In the last lecture we talked about hydroxychloroquine and chloroquine in English as well as in Bengali. Please check that out. Today's topic is compassionate use of Remdesivir for the patients with severe COVID-19. Before that, if you are new to my channel, then please hit the bell icon and do subscribe to our channel. What is Remdesivir? Remdesivir is an antiviral or the medication developed by the American biopharmaceutical company Gilead Sciences, and it is a nucleotide analog, specifically an adenosine analog, which inserts into viral RNA chains causing their premature termination. It is being studied during the 2020 as a possible post-infection treatment for the COVID-19. Remdesivir, a nucleotide analog prodrug, it inhibits the viral RNA polymerases as shown in vitro activity against the SARS-CoV-2. You can see the structure of the Remdesivir is like this. Remdesivir is an antiviral drug. Okay, I have been talking about the hydroxychloroquine, which is an anti-malarial drug. People are talking about how an anti-malarial drug is going to cure an viral infection, but we saw the probable mechanism of that too. So it is an antiviral drug that was developed by the Gilead Sciences in the 2014 as a possible treatment for the Ebola Hemorrhagic Virus or EBV and Marburg Virus infection. Okay. Now we are concerned about the Remdesivir and its probable mechanism of action to treat the severe COVID-19. It is a nucleotide analog, specifically an adenosine analog, evidenced to have broad spectrum activity against the single-stranded RNA viruses such as the coronaviruses. What is compassionate drug use? It is the use of a new unapproved drug to treat a seriously ill patient when no other treatments are available. Here the Remdesivir is considered as a potential repurposed drug considered or the candidate for the COVID-19 or the SARS-CoV-2. Remdesivir is a drug that was initially designed for the treatment for the Ebola Hemorrhagic Virus, I stated just now. And it was during the 2014 to 2016 during the West African Ebola virus epidemic on the people who had the disease. The trials were under the United States Army Medical Research Institute of the Infectious Diseases and the Gilead Sciences. Research with the Ebola virus indicated that the Remdesivir had a very low efficiency against the virus because it causes mutations in the rhesus monkey hepatitis virus RNA replicase, and which causes the partial resistance to the virus. This drug failed during the Ebola outbreak. Okay, as the Ebola virus got resistant due to some sort of mutations taken place in the rhesus factor monkey. So that is why the Remdesivir did not work well in controlling the Ebola during the Ebola outbreak. And these mutations make the viruses less effective in the nature, and the researchers believe that they will just not likely to persist where the drug is not being used. Current research trials of the drug on the COVID-19 has revealed that it is effective against certain key enzymes of the SARS-CoV-2 that cause the COVID-19. And according to the research paper published on the 13th April 2020 on the Science Daily by the University of Alberta Faculty of Medicine and the Dentistry, the scientists have shown that the Remdesivir is highly effective in stopping the replication mechanism of the coronavirus that causes the COVID-19. The finding follows closely on the research demonstrating how the drug works against the Middle East Respiratory Syndrome or the MERS or related coronavirus. So here you can see that the Remdesivir was created and developed by the Gilead Sciences and as a treatment of the Ebola virus, as I stated, and it is a broad spectrum antiviral medicine. And Gilead Sciences subsequently discovered that Remdesivir has an antiviral activity in vitro and against the coronavirus family, and it was previously used in the Ebola against the Ebola and has shown promise in the animal models for treating the Middle East respiratory syndrome MERS and SARS, which was caused by the other coronaviruses. So Gilead began laboratory testing for the treatment of the COVID-19 using the Remdesivir in the January 2020, since it has shown to be an active against the SARS and MERS using the animal models. Okay. Indian drug makers began the developing Remdesivir world's best shot against the COVID-19 in a trial involving monkeys. Early treatment of the COVID-19 with the Remdesivir shown has to reduce the symptoms of the disease and lung damage. South Korea's top coronavirus experts said that the time frame for the effective COVID-19 treatment may be much shorter than what is needed to develop a vaccine, and single out Gilead Sciences Remdesivir is a hopeful candidate. Now coming to the mechanism of action, that is how the Remdesivir works. As you can see, here is a detailed diagram and here is something written, and Remdesivir is a prodrug that metabolizes into its active form GS441524. You don't have to remember this. An adenosine nucleotide analog GS441524 interferes with the action of the viral RNA-dependent RNA polymerase and evades the proofreading by the viral exoribonuclease XON, causing a decrease in the viral RNA production. Though in some viruses, such as the respiratory syncytial virus, but not the Ebola virus, it causes the RNA-dependent RNA polymerase to pause. Its predominant effect is to induce an irreversible chain termination. Unlike with many other chain terminators, this was not mediated by the preventing addition of the immediately subsequent nucleotide, but is instead delayed, occurring after 5 additional bases have been added to the growing RNA chain. You can see here the SARS-CoV-2. Here is ACE2 receptor, which I discussed about the hydroxychloroquine during that time, and the SARS-CoV-2 enters inside the cell, and after entering, the transcription is blocked by this enzyme. First of all, the Remdesivir is a prodrug and GS441524 is an active molecule. This molecule converts into this form, okay, an active form. In this form it is unable to work, so in this form it changes its some modification occurs and then it is able to work in the form of this molecule, okay, and then ultimately the genome replication is halted. Okay, I'm going to show you in much more details in a bigger picture, in a more simplified way. Before that, you must know something. The polymerase enzyme stops adding more RNA subunits, stopping genome replication. These mechanisms are linked to the Remdesivir ability to metabolize into an active form known as GS441524, which is an adenosine nucleotide analog. I told you this is an adenosine nucleotide analog, which mimics the normal adenosine, which is a normal base which is present inside our DNA. The GS441524 interferes with the action of the viral RNA-dependent RNA polymerase and evades the proofreading by the viral exoribonuclease. This decreases the viral RNA production. Let me show you a more simplified diagram, and which I hope it will help you to understand this mechanism more thoroughly. So, it is the RNA of the virus, which is the brain of the virus or the key factor. As it infects the human cell, this is a human cell, okay? It releases its RNA inside the cell, inside the host cell. It goes inside the nucleus where our genetic material here, this is the DNA, is also present, where lot of mechanism goes on, such as DNA replication, DNA transcription and translation, and the modification of the RNA also occurs here. This is the brain of our cell, okay? DNA looks like a ladder. As you can see, the DNA looks like a ladder. It has 4 bases. As you can see, this is A, this is G, this is T, and this is C. Here, A for adenosine, G for guanine, and T for thymine, and C for cytosine. The single-stranded RNA also has the following 4 bases, but here, except of thymine or the T, it has U, the uracil base. The RNA is now inside our nucleus. It utilizes our body's proteins of our cell and gets itself duplicated. That is, the replication occurs. Replication means making the new copies or many more copies of the RNA that is generated utilizing an enzyme called the RNA-dependent RNA polymerase. Okay, this is a bigger picture of the nucleus. From this, you imagine this picture. Okay, so our cell has the DNA-dependent RNA polymerase. We know that. Our cell also, that our cell normally use for the RNA from our DNA. For this, you need to understand a simple thing or a molecular mechanism. This is the simplest way to explain the molecular biology, that is the central dogma of the biology. Here you can see it is the DNA. From the DNA, after transcription, RNA formation occurs. From the RNA, it comes out from the nucleus in the cytosol and makes protein through the process of translation. This is known as central dogma. For this replication to occur, the DNA of our cell uses the DNA-dependent RNA polymerase for its replication process. But when the viral RNA enters in our cell, the RNA-dependent RNA polymerase is utilized. The virus needs the RNA-dependent RNA polymerase, which is also present inside our cell. If the normal thing happens, then it's okay. This process occurs inside our cell, but the central dogma is halted here. The virus utilizes our body's RNA-dependent RNA polymerase, okay, to synthesize its own copies of the RNA or the viral RNAs to make number of copies. You can see from this many number of copies are generated or made out, and these are then transferred to the cytosol, and finally they will make the viral proteins, that is, they will make coat proteins or the spike proteins and package the RNA inside a new virus particle. By this process, the replication of the lytic cycle occurs, that is, it is a process of giving birth or developing the new viral particles. Now, what is the function of the remdesivir? Now, remdesivir can mimic the structure of the adenosine, which looks like a viral adenosine. It can mimic the structure, okay? So, remdesivir is modified into a protein, and which is an adenosine analog, as I said multiple times in this lecture. So, whenever there is a need to be placed adenosine. Here you can say, here adenosine, look this picture now. In making of a new viral RNA, in the process continues. Now you can imagine the new viral RNA process replication is going on. Then the modified version of the remdesivir is then added, and this transcription, the synthesis of the RNA is prevented or halted or blocked. Okay, after this analog is attached. Why? The moment the addition of this analog occurs, or the RNA dependent RNA polymerase cannot transcribe the viral RNA, as it cannot recognize this as its own RNA or the own adenine. It fails. So, got it? Inside it fails to replicate. Inside our cell we have the proofreading activity too, which means fixing out the correction or the mistakes or the errors or the defects we made for a text or something else, for writing, etc. This proofreading activity recognizes the faulty base and cleaves out the base out in general. But in this case, once this adenine is added, or the proofreading activity is of the RNA dependent polymerase is also blocked. Normally it is present in the DNA dependent polymerase for the correction of the errors or the or the mistakes or the mutation occurred or the faulty bases, but here it is in the same thing will be occurring inside the RNA dependent RNA polymerase, but due to this analog or the adenine analog or this molecule, it will be blocked. As well as the RNA dependent RNA polymerase cannot initiate the elongate the RNA further into the new chain of the of the nucleotides. So the virus cannot replicate itself. Okay. So you can see, I want to conclude this process again by saying some points. The research of the scientists of the Gottes lab found that the use of the polymerase enzyme extracted from the coronavirus of the MERS nothing that the enzymes can incorporate remdesivir into the synthesizing single-stranded RNA. So the remdesivir resembling as an RNA building block becomes a part of the new RNA strand. It was noted that adding remdesivir, the polymerase enzyme stops adding more RNA subunits, stopping the genome replication in a simple way, in a single word. And these mechanisms are linked to remdesivir ability to metabolize into an active form known as the GS441524. Which is an adenosine nucleotide analog, I said. Thus, this GS441524 interferes with the action of the viral RNA-dependent RNA polymerase. So, the virus cannot replicate itself. And as we saw using the Ebola virus studies, it was noted that the prodrug remdesivir inhibited the action of the RNA-dependent RNA polymerase, causing the elongation of the synthesized chain. And also the studies are still ongoing to understand whether the prodrug terminates the RNA chains or causes the mutations in the RNA or not. And so cannot in this. So at last you understood that the virus cannot give birth or develop new virus particles. They cannot replicate. So it is a real-time event. During the MERS, which was more lethal, and the SARS-CoV-2, the death is only 3 to 4%. It is not so much lethal as we know. So in the hydroxychloroquine, we saw the idea was to prevent the virus particle to release its RNA. Remdesivir job is to kill the virus after it has begun its infection, okay, by preventing its transcription and making more and more viral particles. So as it worked against the SARS and MERS, it is assumed or believed that it will also work against the COVID-19 or the novel coronavirus. So as they belong to the same family. Thank you. If you like this video, then share and subscribe among your friends and colleagues. Have a nice day.